Good afternoon, everyone. I'm Sharon Rounds. I'm the PI for Advanced Rhode Island CTR, and every month we have a virtual clinical translational research seminar, um, and uh, with alternating talks from distinguished investigators from our collaborating institutions, alternating with talks from our uh, talented awardees. So today we're really, really pleased that Angela Slitt is joining us. She is the chair of the Department of Biomedical and Pharmaceutical Sciences at the College of Pharmacy at the University of Rhode Island. She's expert on chemical exposures, and she's going to talk to us about her work integrating a big pharma approach to mitigate forever exposure and enhance elimination in humans. So, with all of the interest in planetary health, both at Brown and at URI. This is a very important and timely talk. So take it away.
Thank you, Sharon. And I do fully agree that I do think this is a really wonderful area for both URI and Brown and Rhode Island investigators to collaborate on for sure. So just speaking from my group at URI, we are always happy to collaborate. So today, really, what I'm going to talk to you about is work that we've been able to achieve through our URI Harvard P42 funded Superfund Center. This really reflects work done by some very talented trainees. Juliana Agudelo Areza is currently a postdoctoral fellow. She was a PhD student, so you'll see some work from Juliana. Emily Kay, who's actually now a scientist and consultant for Gradient Corporation. August Gendy, who's a doctoral candidate, and Sang Ru, who received his doctorate, was a full-time Pfizer employee and now is at Eli Lilly. So very thankful for trainees. We work a lot with Reiner Lohmann's group out of the Graduate School of Oceanography, and Jitka Bekanova and Simon Vochta work hard to help us create really highly rigorous and refined mass spectrometry techniques that range all the way from cell-based assays to human samples. And lastly, with Harvard, with Elsie Sunderland's group at Harvard, who's worked with us on extractable organic fluorine, Fabian Fischer, who is now an assistant professor at the University of Rhode Island, and Heidi Picard. And really, this has represented a partnership with GSO, the College of Pharmacy, and Harvard School of Public Health. Excuse me. I'm still recovering from a respiratory infection. So today I'm going to talk to you about per and polyfluorinated alkyl substances, or PFAS. And PFAS, are a concern because they're highly persistent. There are easily hundreds in use, but there are thousands being detected. At the time we first wrote our first Superfund, it was probably in the high thousands, maybe 9,000. And every time we write for this grant, it's higher and higher, where it's exceeding 12,000 being detected. So the concern is you have this really large chemical library in which there's not very much known about their human health effects. So when we're talking about PFAS, we're talking either about our ultra short, or long PFAS. So these refer to the carbon fluorine, to the carbon length. So for example, this is a carbon, 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 carbon. They're defined by these carbon fluorine bonds that make them very resistant to breakdown, either by the body or even in the environment. It can take upwards of 900 degrees Fahrenheit to break down PFAS. And then, so we're looking at, here's like a C4 because it's got 1, 2, 3, 4. So this is PFBS. We often talk about C8, which is PFOS or PFOA, and these have 8 carbons and different head groups. So this is a sulfonic head group here. So there are 6 PFAS that have regulatory standards for drinking water in the United States. And numerous PFAS have been associated with adverse health outcomes, in particular PFAS, PFHXS, and PFOA. So these are dubbed forever chemicals, and the reason why is because they possess a long biological half-life, and that really is the challenge for this class of chemicals. It's a challenge for regulating them. It's a challenge for how to deal with exposure and exposed populations, whether that's humans, animals, um, soil and systems. They're just very, very persistent. And in humans, they have a very long half-life. This half-life can range, in the literature, it goes from anywhere from about 1.5 to 12 years, depending on which PFAS it is and which study. So interestingly, PFAS are nearly 100% absorbed in the gastrointestinal tract. So when we think about it, what you take in often through drinking water, sometimes through food, is almost 100% bioavailable. So when we think about this in a pharmacy context, drugs, you want drugs to have high bioavailability. And here you have a class of molecules that are definitely exceeding kind of the capacity that, like exceeding the expectations for a drug, if you will, and that they're very, very well absorbed. So that's the first problem that we're encountering is how do you deal with a class of chemicals that are highly absorbed, is when we're looking, say, for example, in rodent models. They're resistant to enzymatic breakdown, so they get highly absorbed. They travel to the liver, and in the liver, they're very resistant to breakdown. Or we have ones that are newer and they're engineered in such a way that they are somewhat broken down, but then they're broken down to something like PFAS, which is very resistant to further breakdown. And then they undergo extremely slow and poor elimination. So once they're in the liver, a lot of the chemicals are staying in the liver. Some can get excreted back to bile, and then they undergo enterohepatic recirculation. And a small portion gets excreted into through the kidneys into urine. But really, for many PFAS, especially our legacy PFAS, which are over 500 molecular weight, are poorly excreted into the feces. So the problem that you have is that there's a very strong and profound enterohepatic recirculation for many PFAS, especially the ones with a long half-life. So really, there's a lot of gaps in our knowledge, and what we tried to do is at least try to start disentangling some of those gaps from a ADME PK standpoint. So, for example, we know that in the blood we have some unbound PFAS, and they're going to undergo passive permeability. So we're trying to understand first, what does permeability look like for different PFAS? What are the drivers for that? And then next, we know that PFAS can bind to albumin, at least some. And the question really was, how profound is albumin binding? Is albumin a key molecule? And for that unbound PFAS that leave albumin, how do they get into liver cells? Do they use transporters like OATs and OATPs? Do they use fatty acid transporters like CD36 because they look a lot like fatty acids? Once they're in cells, especially liver, what happens? How much do they bind to human tissue? And how profound is that binding? Next, how do they leave and go into bile? And what transporters do they use once in bile? So, for example, do they use transporters like the breast cancer resistance protein that we know is important for excreting chemicals into bile? So these are kind of the different molecular mechanisms that we went after trying to better understand. And really the bigger question, because we can think about this in reverse from drugs, is can we do things to disrupt enterohepatic cycling and mitigate PFAS in impacted individuals? As an investigator all the time, through our Superfund Center, we have questions from departments of health. We have individuals find us at meetings and come up to us and say, I have been impacted by PFAS. What can I do to help? And currently there is no accepted treatment for PFAS mitigation in humans. There are some potential therapies. We're working on that. But really what we're trying to understand is if we understand the mechanisms, we can be more rational and intentional in how we develop therapies. So here this shows a person drinking water. That person is then, that water has PFAS. That PFAS is absorbed. We suspect that maybe OATP2B1 is a transporter, and that's participating in getting PFAS absorbed by the gastrointestinal tract. PFAS then get to the blood. They can bind to albumin. They bind very well to albumin. So really the next question is, how important is albumin, and do we need to think about albumin for inter-individual differences in excretion in PFAS? Next, PFAS go to tissues, and we've worked to really characterize tissue binding and show that tissue binding is really critical for dictating how long PFAS are going to reside in the body. This is important for us to figure out which PFAS, if we have to tolerate or want to use PFAS in a commercial way, which PFAS are more or less likely to bioaccumulate than others. Next, how can we understand and interpret what's going on in the liver? How do things get in? What makes them stay? And what makes them be excreted? And so can we disrupt this cycling? So that's the overall arching goal of the work. Can we decrease absorption through the gastrointestinal tract? And can we enhance elimination by trapping these acidic PFAS into the intestine and increase excretion? So really what we're trying to do, being in a college of pharmacy, so here's Avadesian Hall, is we're kind of trying to integrate common tools that are used in pharma for PK/PD predictions to identify key mechanisms using well-vetted predicative tools. And really what we want to do is do the opposite. So can we use the opposite approaches of drug development and find PFAS that are not bioavailable, or at least less bioavailable? Can we kind of triage or tier different PFAS that are maybe less bioavailable, or provide treatments that can interfere with bioavailability? And so this is my pills, and here really what I'm saying for PFAS is, what can we do? How can we draw upon this framework used in pharma to do the opposite? And I just want to point out, like, this is a real concern for communities that are impacted by PFAS. This is an example of a nonprofit project out of Hoosick Falls, where children have blood levels that are very, very high compared to the average population. The average population would have a PFOA level closer to 5 to 7 nanograms per mil, and these are highly impacted populations. So really what we're trying to figure out is, what can we do for different interventions for highly impacted populations? So the goal of our project within the Superfund context is to first characterize liver exposure. What PFAS are really getting into humans these days? Even though some have been banned or like withdrawn, there are more emerging. So what properties and what ones are we seeing in human donors? Can we identify and validate key mechanisms that impact PFAS kinetics and tissue distribution? And can we use our pharmaceutical sciences background to create and identify rational interventions? So with that, I'm going to shift over to a little bit of data that was generated in collaboration was really done in collaboration with our Graduate School of Oceanography by Juliana Agodela Areza. She's now a postdoctoral fellow with Dr. Fabian Fischer, who's an assistant professor at the University of Rhode Island. And the three of us worked quite a bit along with Jika Bekanova and Dr. Lohman's group on this data. So one of the things that we did was we gathered liver samples banked across the United States from several tissue banks from about 2000 to 2022. And this is just kind of all of the data where we measured and developed a PFAS method for human liver that could measure up to 53 PFAS in liver. And liver is a very complex matrix to work with, so that's in of itself very challenging. And so each dot represents a human. So first of all, what I want to point out to you is PFAS. For example, we couldn't find human liver samples that did not have PFAS. So for the most part, every person, I think there was one sample that didn't. We couldn't find matrices for blanks that didn't have PFAS. It didn't matter if it was a rodent, deer, whatever. Like it's very, very hard. So PFAS was very prevalent in the population. as well as other PFAS that have been removed and are not being used in the United States for commercial purposes. So, for example, we're still finding PFOA in human livers, as well as PFHXS. But what really also stood out to us is that these more new unregulated PFAS that are considered precursors are being found in a high proportion of our human liver samples. And the other interesting thing is that 6,2-FTS and 8,2-FTS, which are fluorotelomers, are also being detected in our human liver samples. And so this is just showing some of the ones that we're concerned about. So when we look at this breakdown over time, what we see was that these sulfonic acids, such as PFAS, dominate in the early 2000s. But really, after 2003, and which makes sense with the fact that PFAS was voluntarily withdrawn by 3M, is that we see now PFAS levels are declining in our human liver tissues over time. So, I think one of the impact of this data, which the paper just got accepted to ESNT this week, is that regulations work. So you take something off of the market, it's going to take time, but human exposure went down. The concern for us is that while PFAS is going down, other more the other unregulated PFAS, and such as sulfonamides and fluorotelomers, the profile in those samples that we did detect PFAS, it's going up. So that's really the concern. So really what we're looking at is we've got really coming back to this slide. Where did this go? We have a lot of different PFAS to tackle. We don't know a lot about their human health effects, especially, say, for example, 6,2-FTS, 8,2-FTS, and these precursors. We know that these precursors can also break down to PFAS. So the concern here is that we have many PFAS. Humans are getting exposed to new ones. We do not know a lot about them, and we don't really even know their propensity for bioaccumulation. But our data is suggesting that they are. And so really what we're trying to do is tackle how can we learn about these molecules and understand their propensity for bioaccumulation in humans, and what can we do to solve that bioaccumulation. I just want to give these examples that when we look at collection year and time-weighted averages, fossa and me-fossa do not decline over time. So these are concerns, is that in our sample set, some things decline, but other things do not. I want to add to this. When we went and looked with the Harvard group with extractable organic fluorine, we thought that a lot of the extractable organic fluorine that we would detect in human livers was targeted PFAS. But it turns out that when we look at that this EOF signature for 2 donors, or a grouped average, we see that there's a lot of EOF in these livers, but it's not explained by our targeted PFAS methods. So this really leaves us to kind of surmise 2 things. One, are there PFAS in our methods that we are not capturing but are being accumulated in livers? Second of all, are human livers also potentially accumulating fluorinated pharmaceuticals? And can we even just explain some of this potentially from pharmaceuticals that the person was on? But the important part is that we have to connect EOF back to really what that is, and whether or not it's PFAS is not completely clear. We do have some other data to suggest that it's beyond targeted PFAS. All right, so far it seems that PFAS are present in human liver and more than the ones that we know a lot about, such as PFAS, PFOA, and PFHXS. So what are the driving mechanisms for retention in vivo? And then can we interrupt those mechanisms for treatment? So I'm going to just present this slide for a minute and talk a little bit about what we can learn from our friends who are doing pharmaceutical development. So I spent a lot of time with Pfizer scientists because I'm a professor in the College of Pharmacy. And along the way, probably like 10 years ago, Eamon Elkatin, these are both adjunct professors, Eamon came to me and said, I want to run a class on transporters. Can you help me? I want to deliver it at URI. And I said, okay, let's figure out a way to do it. And Varma was another scientist who got involved. And so we actually delivered a transporter class to industry scientists. And in that, I would sit on these lectures all the time where they would talk about predictive science. And they created a framework so that basically industry scientists could run a few assays cheaply and then understand whether or not those where their new chemical entity was going to get eliminated. Was it going to be renally cleared? Was it going to be cleared in the bile? Like what transporters were involved? And so Eamon and Varma and a few of other scientists created this extended clearance classification system. And how this works is that you bin molecules into either being highly permeable or low permeability. And this is done with a permeability assay. There's different types, but basically you categorize the molecule into low or high permeability. You look at its extent of metabolism, and then you look at molecular weight. So PFAS really fall into they're mostly class 3B. They are low permeability and they're greater than 400 molecular weight. Now, our newer PFAS, which we know some of them are renally excreted, are less than 400 molecular weight, but they also have low permeability. So in our publications so far, we've done things where we've tried to map out this classification system with PFAS. So this brought us to Sang. So Sang worked at Pfizer doing these assays, and Pharma said, hey, why don't Sang wants to go back to school. Would you like for him to work on this project at Pfizer and run some assays for you guys, and it will help you, and maybe this ECCS class could fit in and work. So what we did was we created a list with Sang of ones that we knew we were seeing in the environment, in our Superfund Center, or in human serum samples, and created this list. So this list was really from our community engagement core, giving us information about what they were seeing in well water, as well as our chemical kind of detection core saying, oh, we're seeing this in water, we're seeing this in our other like bird samples. So this is, and then also with our human work. So Sang went ahead and started screens with these chemicals and then ran them through a bunch of assays, tissue binding, permeability, OATPs. So these are transporters important for liver and for uptake, and then our BCRP for elimination. This work's been kind of further conducted by Olga Skendi, who's a current trainee. So I'm just going to take you through a few of the findings. We have papers in this area, so I'd encourage you to look if you want more information. So really the question is like, does albumin actually matter in vivo, given there are in vitro findings? And really we care because there are diseases that are associated with hypoalbuminemia, and also can albumin sometimes be a reason for reverse causality in these PFAS studies. So Sang did some work with PFAS, and this is just kind of a summary figure showing that when he looked at plasma binding, whether it was human or mouse, there's a high concordance between the fraction unbound for PFAS, so this is kind of what remains, and half-life. So the lower the FU value, that means the stronger the binding in the assay. And so as binding gets stronger, the number of half-lives that PFAS is associated with in humans is much longer. And so both humans and mice were very concordant. Sang was also able to show that, within the species, there's high concordance. So going forward, we said, okay, well, that's really interesting, but we really need to show or understand what happens if you eliminate albumin, and then kind of ask questions about binding. So we start, essentially what went from in silico work to in vitro work done by Sang, ultimately was tested by Olga Skendi in vivo. So Olga did study designs where we used mice that either have or lack albumin. She designed a cocktail in which they receive a 0.1 milligram per kilogram dose of PFAS, PFBS, PFHXS, and 6,2-FTS, and then collected blood over time, and then used a LC-MS assay to basically quantify that. So tissues are basically bloods and tissues are collected over time. So we clearly see with albumin deficiency that PFAS concentrations in the knockout mice, clearly the PK profile changes. And this kind of argues for the importance of albumin for PFAS. We see this similarly for PFHXS. At the time of necropsy, we were able to still detect some PFHX concentrations, and we see that in the knockouts that PFHXS in liver and kidney is lower. So these animals basically their serum profile decreases, they're not holding on to as much, and their tissue concentrations also go down. This makes a lot of sense with what we know about PFHXS. It seems to, it resides more in blood and seems to kind of be bound to albumin. So albumin loss would then change that profile. And we've gone on to do this for other PFBS. I don't like PFAS. I don't have the time to show you, but what I can tell you is we're actively modeling it. And Fabian Fischer has generated some really interesting, nice models that we've been able to so far publish one showing really the importance of albumin for blood, for basically changes in blood, um, partitioning to liver, and then kidneys, et cetera. So he's done really nice predicative work, for example, looking at how you can model changes of plasma concentrations of PFAS in a wild type mouse or an albumin knockout mouse. And that's been a really important addition to our Superfund Center. So far, it looks like albumin is really important. This is great justification for use of albumin in vitro screening, high throughput screens to just understand like persistence of PFAS. And then we moved on to really OATP2B1, and it's important for PFAS absorption. And so really what we want to understand is, could we apply these tools for ADME? And really, ultimately, like, can an apple a day keep PFAS away? And this comes out of data that was done by apple juice and fexofenadine. So apple juice administration is known to inhibit the gut absorption of certain drugs that are OATP2B1 substrates in the gastrointestinal tract. So it's an uptake transporter. It transports drugs from the intestinal, like into the intestine. And fexofenadine is one of those drugs. And we know in humans that if you give apple juice, that fexofenadine bioavailability goes down. This is an established interaction. And apple juice is established and known for certain drugs that it will decrease the bioavailability. So really, how does PFAS relate to drugs and drug development? So the question first really is PFAS or other PFAS, are they substrates for OATP2B1? And are there components of apple juice, or can apple juice inhibit OATP2B1 that then prevents PFAS absorption by the gut? It's known that plant polyphenols have been identified as human OATP2B1 inhibitors. And we know that inhibition of human OATP2B1 can decrease drug bioavailability. Just want to see how we're doing on time. Okay. So first of all, we were able to establish that some PFAS are OATP2B1 transported by OATP2B1. I'm showing you some of the data. These assays can be tricky because PFAS are really sticky and there can be false negatives. But from the data that Sang ran, that we felt confident that PFBS and 6,2-FTS are substrates for human OATP2B1. So this is an uptake ratio. This is showing a comparison of uptake between a transfectant that has OATP2B1 over wild type. So anything that meets the criteria of 2 over 2 suggests that it's a likely substrate. And then the transport is inhibited by a known inhibitor, which is RIF-SV. So both of these, the transport was inhibited by RIF-SV. Other groups have shown this for rat, and I do believe that Bruno Hagenbuch's group is also going to be showing this, but probably better because they figured it out better. I'm showing that other PFAS are also substrates. So from there, the hypothesis was that administration of OATP inhibitors such as apple juice or rifampicin will inhibit OATP2B1 in the gut and either reduce or delay PFAS absorption. And so we went from this in vitro assay to then moving on to mice, where we either administered apple juice or rifampicin, and then looked at how that affected PFAS absorption. So this was work done by Olga Skendi. Olga's a fourth-year PhD candidate. Um, Her ability to get mouse studies done is impressive. And I sent Olga off to go get apple juice. And I really would also say that Juliana really started this. Did the initial study, and I told her I didn't believe it and that we needed to go back and do it again. So Olga's now done this multiple times. Olga literally went to Stop and Shop and got apple juice concentrate. This is the concentrate that we administer to mice, and this really is not a new idea. Other groups in the DMPK area administer mice basically an apple juice concentrate. This was just ChatGPT's version of Olga going off to buy apple juice. That was silly. So in this study design, the mice get a pre-dose of apple juice concentrate or PBS vehicle. We wait an hour. They got a single dose of PFAS, excuse me, either at 0.5 milligrams per kilogram, either in PBS or again within apple juice. And then mice are euthanized and tissues are collected at 2 or 5 hours afterwards. And we then measure PFAS by mass spec. And again, we were surprised. I was like, wow, we were able to reproduce this. So we did see that the apple juice concentration, the apple juice was able to lower PFAS concentration 2 or 4 hours in blood. We were able to see this also in liver and in kidney. So here in liver, the apple juice lowers the amount as well as in kidney. And just noticing here that if you look from 2 to 5 hours, we're still on the uptick of the PFAS being absorbed. So we wanted to kind of get that as we're absorbing. So that's one question. But we said, well, that's great, but we really need to show that it enhances elimination. So we're going to design this differently. So in this case, we moved on. We developed a PFAS cocktail of multiple PFAS and gave a single dose of PFAS, but then gave 2 doses of apple juice, concentrate or vehicle for 7 days over time, and then collected our PFAS. We collected blood and tissues after 7 days. So mice received 1 dose, but multiple doses over time of the apple juice concentrate. And we really saw some phenomenal and interesting things. First of all, as usual, we did see that the PFAS that the apple juice did delay the absorption of PFAS, whether it was PFAS, PFOA, PFBS, or 6,2-FDS, or PFHXS. In 7 days, we did see that both PFAS and PFOA were significantly reduced. So just the apple juice itself decreased that elimination. And so, all right, so I want to move on. So then looking at so the question became, well, that's really great, but we really need to make this a more real world scenario where we're dosing multiple doses of PFAS and multiple doses of apple juice, and then collecting 7 days later. So this is a replicate study. One was single dose, some mice received the multiple dose. So noticing here that the multiple dose mice had higher serum PFAS compared to the ones that received single dose after 7 days. And these guys received repeat doses of apple juice. And again, we saw a reduction in the PFAS concentration. So this was really, really exciting. We feel like we've been able to produce this multiple times. We're in the process of trying to understand what it's in the apple juice that's able to be protective. We did work with Carmen Masirlian, who's an epidemiologist was at Harvard. She now is actually a CEO for her own company called V. And at the time, Carmen and her graduate student mined NHANES data, and we did observe that adolescents consuming pure fruit juice more than once in the past 24 hours had a 15% reduction in serum PFOA concentrations compared with adolescents that did not have any intake. So this was really nice data to kind of support some of our research findings. So last that we were like, okay, this is really interesting. And along the way, we felt as though we also needed to try a known inhibitor. And so we used rifampicin. So in this treatment design, we gave rifampicin As a pretreatment or a vehicle, you give the cocktail with rifampicin. We then look at 2 hours and do a necropsy, and then again do tissue measurements of PFAS. So in this, what was kind of interesting was that we did see a reduction in serum PFBS and PFHXS. And really our concern was just that the rifampicin is what we know is that it's actually less of a it is not a great inhibitor of OATP activity at the gut. However, it is a very good inhibitor at the level of the liver. And that is what we were able to observe. So the mice that received oral rifampicin actually had much lower levels of PFAS, the all PFAS that we measured in their liver at 2 hours. So that was actually really interesting. It really suggests to us that OATPs in the liver are very important for getting those PFAS into the liver. So in conclusion, apple juice extracts, or sorry, concentrate can decrease PFAS absorption in mice. We know that oral administration of an OATP inhibitor is important to decrease PFAS absorption, and it looks like blocking OATPs could be a good molecular mechanism to decrease PFAS absorption. And lastly, coming back to the initial part of the story, it does appear that albumin is a key factor for serum retention, and that's something to consider in screens and inter-individual differences. So coming back, we believe that this is important to tackle. We're currently also working kind of in the area of excretion, for sure. We do have a study that's been funded through the Department of Defense that is looking at military service and then veteran status. So, for example, we are enrolling, we are almost to the point, we think in several months we'll be able to actually launch the study. The goal of the study is to recruit 500 US veterans and veterans who then have serum PFAS level that exceed 20 nanograms per mil, which is kind of the cutoff from the National Academies of Science and Medicine for treatment. So if you exceed 20 nanograms per mil of key PFAS, you will then be invited to be part of a study in which you can take colesevelam, which is a bile acid sequestrant, and you can take that colesevelam for 12 or 24 weeks and have your blood remeasured for PFAS concentrations. So this study, we're really excited to work with the VA in Maine, as well as the Robley Rex in Louisville, and our research partners, Rodney Sparks, Matt Cave, and Barbara Furman, to be able to really identify a intervention that's feasible and effective and affordable in the VA population. So with that, there's so many people to thank. Dr. Fabian Fisher at the University of Rhode Island, Slitt members past and present, Juliana Agudelo worked tirelessly on the methods, Olga Skendi, my current students, former students such as Emily Kay and Sang, our steep collaborators like Reiner Lohman and Yitka and Simon, Elsie Sunderland, Laurel Shader at Silent Spring. Our steep collaborators are tremendous. It's a great, great, great program. Eamon and Mithena Varma, who are our Pfizer colleagues who allow us to fund students and use their budget. University of Louisville, it goes on. Thanks to NIEHS, the Department of Defense, and for all of you for listening.
Wow, that is spectacular. Very, very, very interesting.
I hope I didn't talk too fast.
No, perfect, perfect. So it raises so many different questions. First of all, how can a citizen determine whether there are dangerous levels of these chemicals in our drinking water? Some, like either city water or well water, whatever.
Well, so I mean, first of all, currently, there was EPA guidance for drinking water. So with that, many water systems are currently monitoring and trying to achieve the EPA water standards. So I think the good thing is that for key PFAS that we know are bioaccumulative, accumulative, public water systems are monitoring. Now, if you're on well water, there you have a few options. There are private testing companies that will do water tests. You can go to EWG, and there are some water tools through Environmental Working Group, which you can plug in your zip code. And in that zip code, it will kind of tell you if there's been PFAS detected in your area. And lastly, prophylactically, you can always use, carbon filtration will remove many, but not all PFAS. So Brita filters, for example, you can go to Steep's, our Steep Superfunds site, and we have tools in which, you can look up and they will tell you what filters you can commercially buy that are available and what PFAS they remove.
Wow, very interesting.
Yeah.
And are these environmental standards being degraded nowadays?
You mean because of the political climate?
Yeah.
I mean, I do believe that there's conversation around changing those. I don't know the status of that.
Hmm.
But what I would just say is that we do know that and there are certain tools for that you can use to kind of look at your, like if you have water levels of a certain amount, how you might bioaccumulate and how that compares to the human population. So ATSDR has a calculator. And so I would, say that that's also useful. The previous standards were 70 parts per trillion. Those water standards were based off of rodent data. The current data is based off of human data. It's a much more stringent of, I believe, 4 parts per trillion. So that's a little bit harder to, that's harder to achieve.
And we're all wondering, how many apples are you eating per day?
I'm not probably eating enough, but I probably should given all my PFAS work. Um, but I think there is some really interesting rational, like there's a science, like this has a scientific rationale to it. So that's pretty exciting. We're not just throwing something at the wall and hoping that it sticks.
Yeah, yeah.
Um, yeah.
Do other fruit juices have similar effects?
So we tested grapefruit juice and it didn't, but grapefruit juice has a much different pH. So one of the concerns with grapefruit juice is that its pH is much lower and PFAS are acidic. So one concern is that they could basically then be unionized and maybe get absorbed.
Mm.
Um, there's other literature around differences between apple juice and grapefruit juice. They don't have the same exact phytochemical constituents. So apple juice and grapefruit juice are not identical. So the short answer is yes, they could. We haven't, once we ran with apple juice, we've been so busy testing that we haven't tried others.
Wow. And is this patentable?
Oh boy. Um, I think that depends. I think if you could potentially find a molecule that's novel or new, then yes. I think that, a lot of the times there are ways to have trade secrets, and things like that.
Hmm. Very, very interesting. Um, well, and the another question that I hate to be monopolizing this, but I'm not other questions in the chat is I summer in Casco Bay in Maine, and there was a big PFAS spill at the Brunswick Naval Air Station a
Mhm.
couple of years ago. It was a firefighting foam that got loose into the watershed. So the question is, does this, after accidents like that, do these chemicals show up in seafood, either mollusks or fish?
Yes, don't quote me on the amount because I don't know that. I do know that our Superfund group has worked on this a little bit, and I do think that other groups are funded to do this too. So I do think there's some concern. I mean, fish for sure. The question is, the accumulation in the parts of the fish that you're eating versus not
Uh,
completely clear. I'm not a fish person, so I feel bad that I'm probably not giving a totally correct answer. What I would say is I'm certain that groups are working in this area to figure that out, and that is definitely a concern.
So a question from the chat is, uh, are we, it's the finding PFAS everywhere simply because of better tools, or is there more contamination out there in the environment?
Well, I mean, I kind of think it's both. So the
Okay.
tools part is with with high resolution mass spectrometry, we now we have the ability to detect new things, right? So that's Gen X was really found because of the ability to have like untargeted high resolution mass spectrometry. And so I do believe that others, that new structures are being found. But I think the other question becomes, whether or not is there new PFAS or is there more? I mean, we are seeing that there are other PFAS present. Other PFAS are being used in products. So I don't think that there's decreased usage. I think there's decreased usage of some PFAS, especially in the United States and other countries. So it's a little bit of both.
Okay, great. Well, Angela, thank you so much. This was really very, very interesting, and I have to say a little scary. Um, we really appreciate your taking the time and sharing this really interesting work with us, and good luck to you on your nice new grant. That's terrific. I'm really glad to hear that. We write a lot of letters and we frequently don't hear about what the result was. So I hope it was helpful. Anyway, thank you
Thank
again.
you.